Hi, everyone. Welcome today. This is our first HBI 101 talk of the semester, and we're really thrilled to have with us today Dr. Clifford Woolf. Dr. Woolf is the director of the Kirby Neurobiology Center at Boston Children's Hospital, as well as a professor of neurology, a professor here in the Department of Neurobiology, and at the Harvard Stem Cell Institute. And we're also lucky that he's on our HBI steering committee. So in preparing for this intro, I was looking at his Wikipedia page, and despite working with him on our steering committee for a while, I was just amazed by I was really blown away trying to synopsize what he does. But looking at from your own notes, I can help you put together all of that. He studies plasticity in neurons, different types of plasticity, structural, functional, chemical, and understanding that in both adaptive and maladaptive problematic situations, with an interest in pain, nerve regeneration, neurodegenerative diseases. He also has expertise in using iPS cells, looking at patient-derived stem cells to look at drug candidates and to model different disease states. He has about 35 different patents. He's founded several companies. His work is really broad-ranging: cellular and molecular systems, neuroscience. And as I was reading about it, one thing that really stood out was just how deeply collaborative it is, both in the academic community here and the pharmaceutical companies around. So yeah, he grew up in South Africa, where he received his MD, PhD, then went on to become a professor of neurobiology at University College London before coming here to Boston. And with that, I'll hand it over to him. Thank you so much for doing this. Oh, actually,
hi, Parizad. Thanks very much. Thanks for the invitation. It's a great pleasure to have this opportunity to share with you our work that is attempting to both understand and conquer pain. And as you indicated, I see my work as a team effort. When I first started research in those times at University College London, each faculty member had one graduate student and one postdoc, and they kept everything a total secret. And now it's completely different, as I hope I can show you. So as Parizad indicated, I'm the founder of 3 companies, and I sit on the SAB of several others. The only one that is relevant today is a company that was spun out of work that was done in my lab that enables us to measure the behavior of mice in a new quantitative machine learning based way. So I will be mentioning that. So the big issue is why study pain? And frankly, when I first came to the US in 1997, if you looked at the different areas that the NIH funded, pain was right at the bottom. It was the lowest of all. And partly that was because pain was just thought to be a symptom. It was thought to be something that you had to treat the underlying pathology, not the pain itself. And at that time, 1997, was just the beginning of the opioid crisis, when certain companies started quite ambitiously pushing the use of opioids for patients with the claim that patients would not have an abuse liability. And that turned out to be completely wrong. So if one looks at the incidence of pain, pain is extraordinarily high. That you can see that 20% of the US population have chronic persistent pain, and about half of them, or just under half of them, the pain is really of high impact, which means that those individuals typically cannot do regular jobs. It impacts their personal lives, and they really suffer. So it is a major problem. And actually, if you look at the amount of money that is spent on different diseases, cardiovascular diseases, cancer, diabetes, etc., and you look at pain, pain is right there amongst the very top. So it is a major issue. The NIH have now changed their view, and that is largely because of, as illustrated here, the opioid crisis has led to an absolute epidemic of opioid overdose, and that the number of patients who are individuals, who die every year is now exceeding 100,000. So this is this is a total nightmare. Frankly, it makes COVID seem like a little blip relatively. So this is a major issue. And at last Congress, I recognized that, and about 8 years ago, the Congress gave a substantial amount of money to the NIH for the HEAL program, which is to help end addiction long term. And it has two components. One is to deal with addiction, to try and minimize the risk of addiction, and to deal with people who have overdoses, to reduce people who are taking illegal drugs. And the other is to try and conquer pain, because literally, if we can eliminate opioids as a prescription a therapy, that would make an enormous difference. And frankly, that is my prime motivation. So to do that, my lab has two major drives. One is to understand the mechanisms of pain, and the other one, which is more recent, is to attempt to develop novel analgesics that have no abuse liability. And frankly, in the last 5 years, we've essentially changed our lab from being a purely academic research lab to what I call an academic biotech, as I'll illustrate. I'll briefly describe some of the approaches that we are using to understand the mechanism of pain, but most of my discussion is going to be on the platform that we've put together to try and develop novel analgesics. So this is my team. There's quite a few members who are actually in here. It was a perfectly balanced team in terms of identical numbers of male and females until George, who's sitting over there, arrived this month as my newest postdoc. And well, you've upset the balance a little. But this is a great team, and frankly, I just couldn't do the work without their commitment, their motivation, and their enormous technical skills. One of the big breakthroughs in our understanding of pain has been the appreciation that there are multiple different kinds of pain. When I first began working in the pain field in 1979, literally pain was thought to be a single symptom, and sometimes it was acute, and sometimes it was persistent, and sometimes it was associated with injury, and sometimes with nerve damage. But it was always thought that the same circuits were activated and the same mechanisms. And at that time, the World Health Organization had a very simple chart, which they recommended as the best means of treating pain. And that is, if you had mild pain, you took a non-steroidal anti-inflammatory drug. If you had moderate pain, you took a combination of an NSAID together with a weak opioid. And if you had severe pain, then you took a strong opioid. And this was called the analgesic ladder. And frankly, it was a ladder to opioid addiction and opioid tolerance and opioid overdose and all these horrible things. And the assumption of that WHO, the World Health Organization, the ladder was that all pain is the same. It just varies in its severity. And that is completely wrong. So one of the major forms of pain, the pain that all of us in this room hopefully have, is nociceptive pain. That is the pain that is activated by noxious stimuli. And by definition, a noxious stimulus is a stimulus whose intensity is just below the point at which we have tissue damage. And the reason we have this nociceptive pain is that it warns us of danger in the environment. And frankly, we have danger in the environment every day, and we don't even think about it most of the time. Obviously, if we touch something too hot, like a stove, we shout out, should we withdraw? If we, the same with go walking on freezing ice. If we hammering nails and we happen to hammer our fingers, so the point is that there are thermal drivers, there are mechanical drivers, and as illustrated there are also chemical drivers, of which the pungent ingredient in chili peppers, capsaicin, is a great example. Capsaicin turns out to produce its burning sensation by virtue of the fact that it is an agonist for the TRPV1 receptor ion channel, which David Julius discovered, and he got the Nobel Prize for that in 2020. So these are physiological pains. These are pains that we need to preserve, because there are individuals who have a syndrome called congenital insensitivity to pain as a consequence of genetic mutations in key genes that mediate nociception. And these individuals do not get a warning of potential danger. And in consequence, they injure themselves. And if you look at these individuals, the tips of their fingers, almost all of them are gone. Their mouths are severely scarred because when they eat and chew, they don't know the difference between chewing meat and chewing their tongue. And if they have a shower, they cannot differentiate between something that's pleasantly warm and something that is burning hot and will actually injure their skins. So we need that. And these individuals are all damaged, and most of them die in their 20s. So our capacity to feel protective pain is absolutely essential. There are a broad number of clinical pain syndromes. Some of them are nociceptive in the sense that, yes, a scalpel blade cutting through skin will produce nociceptive pain acutely, but then there's tissue injury and inflammation and a whole series of post-operative clinical issues. There's inflammatory pain, which in some cases is acute. It can result from tissue injury, from pathogen invasion, and from a number of diseases, such as autoimmune diseases, diseases like rheumatoid arthritis, where there is a pathological inflammation. Country Jane sitting here has been one of the major drivers in my lab of interrogating how the immune and nervous system work together. And she has discovered novel ways in which these two systems operate to detect danger, to react to danger, but also to pathologically interact in a way that can drive ongoing persistent pain. And then there's neuropathic pain, which is pain associated with damage to the nervous system. Most of the patients with neuropathic pain have damage to the peripheral nervous system, but spinal cord injury and stroke can also cause neuropathic pain. And this is a true dysfunctional pain. There's no, with inflammatory pain, one could argue that the pain helps healing to occur by avoiding contact with an injured or inflamed part of the body. Obviously, that's not really true in a patient with rheumatoid arthritis, where the slightest movement of a joint is extremely painful and the pain doesn't go away. But there are inflammatory situations where one could say the evolutionary drive for inflammatory pain helps healing to occur. But in the case of neuropathic pain, it is totally pathological. It is a malfunction of the nervous system such that we feel pain as a consequence of the damage to the nervous system. And then there's visceral pain. A postdoc of mine who's actually not here, he's visiting his family in Taiwan. Yuting is studying visceral pain, and that is turning out to have many features that are substantially different from the pain that would occur from a stimulation of the somatosensory system, the skin, joints, muscle, tendons, etc. And then there's a class of pain called nociplastic, which there's no noxious stimulus, there's no inflammation, there's no damage to the nervous system, and that these patients have ongoing, typically widespread pain, of which fibromyalgia is one of the typical examples, temporomandibular joint disease is another, tension-type headache, irritable bowel syndrome. And all of these are situations where there's no peripheral pathology, but there's terrible ongoing persistent pain. So we are attempting to understand the mechanisms of pain, and one of the ways is doing that is to model pain in rodents. And this is a major collaboration with David Ginty's lab, who's just around the corner over here. We have a joint brain R1 grant together, and I'll describe a few of the things that we are doing with that. We've also very recently become a part of a consortium that Will Renfeld, who's a neurologist at Brigham Hospital, has put together. And we are looking at human neuroma pain. It turns out that patients have an amputation. Many of them develop very severe pain of the injured nerves. But in the same patient, some of the nerves the neuromas may be painful and others aren't. Some individuals have no neuroma pain, and some have it so bad that they even threaten suicide. And we are now working on those human neuromas to try and identify what features may be the driver of that pathological pain. We are also doing a substantial amount of work on trying to model pain in various disease states using iPSC, human iPSC-derived cells, and Basic Pepova, who's sitting here, is one of the leaders in this. We are modeling with two teams, the Lab of Systems Pharmacology, which is one floor below here, Peter Sorger's lab, working with Caitlin and with Ross Siegel and Dana Farber, trying to understand how chemotherapeutic agents used for treating cancer can damage peripheral nerves and cause both neuropathy and neuropathic pain. Unfortunately, I don't have time to discuss either the neuroma or the CIPN, but just a few words about some of our ways that we're working with the Ginty team to explore the mechanisms of pain. Sunzi Zheng, a relatively recent postdoc in the lab, has been looking at neuropathic pain to try and identify the major features. And he's making some enormous breakthroughs in our understanding of that. And one of the techniques that he uses is to use GCAP6 in dorsal ganglia. That's where the cell bodies of the sensory neurons are located. And he can identify which sets of sensory neurons are activated in normal, healthy situations and which are activated in pathological situations such as neuropathic pain. And this is really beginning to reveal what are the drivers of this persistent neuropathic pain. David Yarmolinsky, another postdoc in the lab, has been working for several years developing what we call a spinal window preparation. This enables us to record from neurons in the dorsal horn of the spinal cord, and we use genetic techniques to get expression of GCAM in particular populations. They can be either interneurons or projection neurons or whatever. And the beauty of this approach is we can record the same sets of cells for over a year. So we can look, instead of the standard approach to studying the activity of certain circuits by studying the circuit for a few hours on one day, we can literally look at those same neurons day after day or week after week and look at the changes that occur in clinical conditions. And this is really beginning to reveal some very exciting data. So, as illustrated there, you can just see the neurons you can pick up, you can stimulate the hind paw. You can do this in lightly anesthetized animals, but also now David is beginning to do this in awake animals. So we can actually interrogate the functional activity of the nervous system in behaving animals. Dan Taub, another senior postdoc in the lab, has been looking at the cortical activity that may be a major driver of the perception of pain. And to do that, he's using optogenetics and as well as DREADS to manipulate different, very specific circuits. And he can identify which areas in the cortex, when activated, produce pain-like features, and those, when inhibited, don't, and vice versa. And he's discovering some very new, exciting features, and just had a paper that appeared a few weeks back in Nature Communications on that. But what I'd like to now do is describe to you the analgesic discovery platform that we've set up. And we are attempting to go after a number of different pains. The major one, which I'm going to talk about, is a means of selectively silencing nociceptors, which are the high threshold sensory neurons that are activated by noxious stimuli. So we are attempting to find a way that we can identify drugs that only act on these neurons and not on any other excitable cells. And in this way, can switch off the activity that triggers the sensation of pain, at least in the setting of trauma and surgery. We're also doing work developing neuroprotective agents to prevent damage to the nervous system that causes neuropathy. That is a major collaboration with Peter Sorger, and that's going extremely well. We also have an assay now to look at the sensitization of nociceptors. When nociceptors are exposed to an inflammatory milieu, their threshold drops such that they can now begin to be activated by innocuous stimuli. All of us who've had tissue injury or surgery or inflammation are aware that suddenly touching that area is painful, whereas normally touching the skin is obviously not. And we can now assay that change in the sensitivity of those nociceptors and use that to assay, to run screens to try and develop new more effective treatments than non-steroidal anti-inflammatory drugs. And then finally, we are also teasing out both the mechanism of pain and identifying novel approaches to manage it. Amazingly enough, this analgesic program actually started in this room, because Bruce Bean and I made a presentation to the Harvard board, and one of the members of the board was Peter Sorger, who's the director of the Lab of Systems Pharmacology. And at the end of our presentation, he came up to us and he said, "Have you guys got a DARPA grant?" And we both said no. And DARPA is the Defense Advanced Research Program Agency of the Department of Defense, whereas Peter has, and he knew them well. And he said, "Are you aware that they've just had a call for a novel consortium to identify novel analgesics with no abuse liability?" And neither Bruce nor I knew about it. And Peter activated us, and we put in a grant just over about 6 years ago, and it was awarded. We now have a consortium that links up Boston Children's Hospital, where my lab is based, and our medical school. And this involves a major collaboration with Bruce Bean, and I'll tell you some of the work that he's doing for us. Peter Sorger, who runs the operation, Jen Smith, who runs the screening core here at Harvard Medical School and has played a major role in our platform. More recently, Mark Namchuk, who is head of translational research here at Harvard Medical School, was recruited a few years back to the medical school, and he's become a key member of our team. He has enormous experience working in biotech, so he brings to us a very useful view of what the pharmaceutical industry expects. We've also, this has been a collaboration with the translational arm of the NIH, NCATS, initially with Ilya Singak and now with Carlos Tristan. I'll tell you in a moment what that collaboration involved. So the major effort that I'm going to talk about now is our attempt to identify and test and evaluate nociceptive selective silencers. And as I've mentioned, this could be used in the setting of trauma. And that's why DARPA, which is part of the DoD, has initiated this program, because they want novel therapeutics that can be used to treat battle wound injuries. If you can imagine soldiers running around with syringes filled with opioids to apply to someone when injured, well, the temptation to them to inject the opioid before the injury is too great. So there is that abuse liability. And in addition, if you have someone who's got a major injury and you give them a very high dose of opioid, they may stop breathing. So there are real problems with the current therapies. So the DARPA are attempting to sponsor us to come up with a new approach. So that trauma would be one, surgery would be another. I don't know if any of you have had a general anesthetic, but when you have a general anesthetic, there are two goals. One is to suppress your consciousness, and the other one is to suppress your feeling of pain. And to do that, you need a high exposure, because the general anesthetics act on most neurons, and you essentially are shutting down many of the activities of the nervous system, such that when the anesthetic is terminated, almost everyone has cognitive problems. They just feel a bit confused, as they can't remember what happened before the operation, and it takes several weeks for them to feel that their cognitive functioning is returning. Whereas if we have nociceptive silencers, we could eliminate the pain sensation and give tiny sedative doses, which would hopefully be much, much safer. And then there are a broad range of pathological pain states that are driven by nociceptors. And if we had a drug that could switch that off, that would be enormous. We are developing drugs that potentially can be given systemically in the setting of major trauma and surgery, and also topically. Again, I don't know if people are familiar with local anesthetics, which are non-selective sodium channel blockers. If you go to the dentist and you get an injection, yes, you don't have pain when they drill, but you also don't have any sensation and you don't have any motor function, because it blocks all the activity of all fibers in the peripheral nerve, sensory, motor, autonomic. And for those of you who had a baby and had an epidural, yes, that reduces the pain during delivery, but it also prevents you from standing up and walking and holding your baby, because it paralyzes your legs. So if we can develop nociceptor-specific silencers, we could eliminate these broad effects of the currently used local anesthetics. So how are we doing this? Well, we've got a number of different approaches. One is, and again, this is DARPA that has pushed us, to take the argument that instead of going for the standard approach of pharmaceutical discovery, which is to identify a novel target involved in the disease, and then identify a drug that acts only on that target. And that, frankly, is 99% of what the pharmaceutical industry is doing now. What we have been attempting to do is to say that you may get much better efficacy and safety profiles if you go for multiple targets, and that maybe there's not going to be a drug that will, by itself, acting on a single target, can be a nociceptive silencer. But if you act on multiple targets, you can get that profile. So that is a big part of what our platform is, identifying targets and testing whether acting on those targets together can have a greater efficacy and safety profile. And then the other is, instead of relying only on preclinical work on rodents, is to introduce human cells as part of our assay system. And I'll describe how we are using human iPSCs as a means to model key aspects of pain and to use them to look for the most promising compounds. But we do need a means of interrogating behavior in rodents, because in the end, in vivo efficacy is going to be a key predictor of whether a particular compound is likely to have benefit in patients. And what I'll describe to you is the way in which we are attempting to move from very crude behavioral assays, which have been absolutely standard for the last 50 years, to a new machine learning-based approach that is providing us with much more sensitive and unbiased information, and is suddenly bringing back the value of work in mice and rats. So a big part of our analgesic drug discovery program was initially to having identified a set of targets, and I apologize for not identifying them here because we still have intellectual property issues, but we have identified 3 different targets and have run massive screens, initially using a Thalim-Flux assay to identify compounds that act on these different targets. And then very recently, Bruce Bean and his team here at Harvard Medical School have introduced an automatic patch clamp device, which enables us to look at detail at the effects of the hits on particular ion channels in terms of their activity, whether it's use dependent, whether it's voltage dependent, and get enormous information on the selectivity of these compounds. In addition, as I've indicated, we are using human neuron phenotypic screens, and I'll take you through some of the aspects of that, as well as this new approach for in vivo evaluation of the efficacy, but also of adverse effects in mice. So in terms of using human neurons, as I indicated, this is a collaboration that has been with the NCATS, the translational division of the NIH. And last year, we published a paper describing a new differentiation protocol that enables us to take stem cells and convert them into sensory neurons. And the beauty of this is that at NCATS, they have a robotic system whereby once the protocol is established, they can use the robots to generate the cells. And they literally are generating millions and millions of cells every week. And they just ship them to us, and we use millions and millions and millions of cells. In the past, my lab spent something like 15 years trying to optimize ways of turning human stem cells into nociceptors. And the best we ever got was a point, and a very nice Nature Neuroscience paper in 2017, where we managed to get a few hundred cells that were nociceptor-like. But now we have millions of cells, which is enabling us to run assays in a way that was just not conceivable before. And the way we do this is in vitro. We take our pluripotent stem cells, we then put them through a various number of steps whereby we can turn them into what we call nocispheres, which are neural crest, early neural crest-like structures that, again, with further steps that resemble the developmental changes that lead to the formation of the central system, lead to the generation of a set of sensory neurons. We don't make a single set of sensory neurons. We make something that looks pretty much like the sensory neurons that are present in dorsal ganglia. So there are a mixture of low threshold and high threshold sensory neurons. There are some that are peptidergic and some are non-peptidergic. So we have a group of cells that pretty closely resembles that which is present in humans. And as illustrated below, you can see by day 35, we get these clumps of cells which really do look like the sensory neurons. If you are able to access a human DRG and culture the cells, it'll look very similar to that. And we've managed not only to make these cells, but to record their activity. And as illustrated at the bottom there is just a recording of GCAMP activity in a plate. Jai-Hoon, who's sitting over here, has managed to set this up and enables us to record from these cells in a 384-well format. And we can really run assays on these cells and identify compounds that have a selective action only on nociceptors and not on other neurons. And not only does Jai-Hoon run GCAMP-based assays, he also can grow these cells on multi-electrode arrays and record from them as well. And illustrated here is one of the assays that he's using, where he's looking at the effect of a vehicle, DMSO, and then a compound that we got from our screens at 3 different doses. You can see that at least at the highest dose, there is a complete inhibition of the electrical activity of these neurons. So we have assays now where if we're looking for nociceptor silencers, we can identify whether we can get that silencing in human neurons. Selwyn, who is one of the program managers of our DARPA grant, has a major role in this. And one of the aspects of the platform that we've set up is not only to look for compounds that inhibit nociceptors, but to run counter screens to ensure that there's selectivity of action. So we run the screens not only on nociceptors, but we make cortical and motor neurons and cardiomyocytes, because we are looking for compounds that are only active on the nociceptors and have no activity on these other cells. And that, again, is a way of trying to eliminate adverse effects upfront, identifying compounds with the desired profile that we're looking for. And this is just an example of one of a set of 4 different hits that we identified, where we've looked at the activity on nociceptors, cortical and motor neurons. And you can see that we are identifying compounds, and we have run screens on hundreds of thousands of compounds, but we've got a handful of them that do have a selective activity in silencing nociceptors with minimal or no activity on the other excitable cells. Once we've identified the most promising compounds, we need to see, do they also act on mouse nociceptors? And Rasheen Powell, a postdoc in my lab, working together with the Bean lab, does this assessment. They take primary sensory neurons in the mouse and make sure that our compounds that we've identified by screening against human targets has some activity on the mouse. And that's important, because if we're going to do in vivo testing, we need some way of making sure that there's no big species difference. And this is particularly important because Vertex announced last year that they have a NaV1.8 blocker, which has efficacy in a number of different clinical trials, but it has no activity or minimal activity on mouse nociceptors. So they have no preclinical data. They are having to explore the potential utility of this compound only in humans, whereas we can do the human studies in vitro with our stem cell derived, but we also can do our mouse studies by ensuring that the compounds that we're looking for do have activity in the mouse. Once we get the most promising hits that have the profile we're looking for, then our medicinal chemistry team, and that's run by Mark Namchuk with Corey Bauer, a postdoc in the lab of systems pharmacology, working with him, and they are looking at the hits and then creating analogs that will increase efficacy, decrease off-target effects, and also have drug-like properties, which are important. The drug needs to be stable. If you're going to give it systemically, you've got to identify whether you can give it orally or by injection. You need to know its PK. You need to know whether it binds to proteins and whether it can cross the blood-brain barrier or not. So what I'd like to end off with is to take you through what I'm most excited about, which is, frankly, at the very beginning of our DARPA project, about 5 years ago, we said that we were going to introduce new means of measuring pain, but we hadn't actually had them running at that time. And I'm really pleased to say that the work has advanced to a point at which we now have not only high throughput platforms for screening against multiple targets, we have human neurons to screen, but we also have an in vivo efficacy platform. And this is enabling us, as I'll show you, to identify not only stimulus-evoked pain. The standard approach to measure pain in mice or rats is you take a stimulus, a noxious stimulus, say a pinprick, or you can use a laser to heat the skin, and you get an ouch response by activating the nociceptors in the mouse. And then the animal withdraws and typically turns to try and bite the stimulus. And that is the typical measure used in trying to assess whether there's activity in the mouse. However, most clinical conditions have ongoing spontaneous pain, and there's no way of measuring it. So we've introduced that. And we can also model clinically relevant pain conditions. Instead of just a pinprick or heating the skin, we can now attempt to model the different kinds of pain that I described, the nociceptive, the inflammatory, the neuropathic, the visceral, etc. And the way we're doing that is with an automated readout using machine vision and machine learning. And this is really enabling us now to have an in vivo platform for analgesic drug discovery. So the actual instrument that enables us to do this was discovered by a graduate student in my lab, David Roberson, who was a PIN student, a programming neuroscience student. And he came up with the idea that if you could just have a freely moving mouse, a black box, mice are nocturnal animals. When you put them in the light, they tend to freeze, they get tense. So if you have them in the dark, they will explore and move around much more. You also have them in a box such that there's no observer. Mice are, again, very nervous. If a human is close to a mouse, it becomes very tense, its cortisol levels shoot up, and you are often just studying stress-induced changes rather than what you really are interested in. So here we can leave the mouse on its own in the dark, and we can measure the movement of the mouse in space, and it's up to the mouse to do whatever the mouse wants to do. And the box is illuminated with near-infrared light, which the mouse can't see, so it's totally black. And then the other great idea that David had was to introduce the capacity to measure surface contact on a glass plate at the bottom of the box. And that's using frustrated total internal reflection. If you put near-infrared light on the side of the glass, it bounces inside the light until there's surface contact, and that'll cause a fluorescent signal to be generated, which can be picked up by a near-infrared camera, and that causes what we call poor luminescence. And that is enabling us, as I'll show you in a moment, to both measure the change in body position in space and time, but also to look at surface contact. And if we're all familiar, if you have pain, if you've stood on a thorn on your paw, on your hind leg, then you avoid contact until healing has occurred. And that is exactly what happens in mice as well. So this is the image of what you can see. The purple color is the surface contact as detected by this frustrated total internal reflection. But you can also see the mouse moving around, exploring, and we can pick that up automatically. This work was published in December 22 with Alex Zhang and David Roberson being the lead authors. Alex Zhang is another graduate student in the lab who just defended his thesis last year, and he's completing 1 year postdoc in the lab just to finish off some projects. He has been responsible for most of the analytic tools, and more recently has been joined by Omar Barkai, another postdoc who's working with him to find automatic ways of detecting changes in behavior. And they're assisted by B.R. Jean. And we have an in vivo team run by Bruna and Mariam, who run all the experiments and essentially are generating all the data. So this is one of the latest ways that we can identify what an animal is doing in space. By the skeleton tracking, we have 51 points. So we can enable us to see exactly what the mouse is doing, how it's locomoting, how its paws are distributed, if it's rearing up, if it's biting, if it's licking, grooming, etc. And this is the data that we get, which Alex and the team can analyze. And it's really amazing. With these 51 points, you can get almost every aspect of a mouse's behavior, but you can quantify it, measure it, and analyze it. We can separate out the surface contact signals from the frustrated total internal reflection from the body surface, and we can superimpose them. So here we've got two video screens, and we can also impose them so we know exactly where we can relate what the animal is doing, in this case, biting and flicking of the paw with the change in paw luminescence. And one of the exciting features of this paw luminescence signal is that it turns out to be linearly related to the surface pressure on the glass plate, so that if you increase pressure on the plate, you get a bigger and bigger signal. So the signal is telling us how much pressure, how much force is being applied to the plate. And as illustrated there, it is dynamic. As the animals walk, one paw is on the surface and the other paw is up. So we get in dynamic, lots of data about the normal locomotion and the balance of activity between the different hind paws. So one of the first things we did was to see whether this approach enables us to pick up analgesic efficacy with high sensitivity. And the way we thought we'd do that was to have a model of inflammatory pain. And we did that using a zynosine injection. Zynosine mimics at least some features of pathogen invasion. And then we used one of the standard non-steroidal anti-inflammatory drugs, ketorolac, which is widely used clinically, which is a well-known non-selective cyclooxygenase inhibitor, which works well in inflammatory pain. And one of the features that we were interested in is that the doses and exposure that produce analgesia in human are about 10 times lower than the doses that are required to develop an analgesia in a mouse. And we always wondered, why is that so? Did that reflect some different metabolism? Or there'd be many potential explanations, but none of them really made too much sense, at least to me. So we started off by seeing whether this fluorescent imaging of surface contact could give us information about the presence of pain, mechanical hypersensitivity, as well as its reduction by an analgesic. So as illustrated over here, we've got the paw luminescence ratio. That's the ratio between the left paw, which has the zynosine injection with the inflammation, and the right paw, which doesn't. And in the control situation, the ratio is 1. If there's identical surface contact on both paws, you then produce inflammation in one paw, and it drops to about below 0.5 because the animal avoids surface contact on the inflamed paw. And then we're giving 3 doses of ketorolac, 1, 3, and 10 milligrams per kilogram, and a saline control, done completely blinded. And that's the beauty about this. You just put the mouse in, no one knows what treatment the mouse has had. The actual analytic package just picks up the change in paw luminescence automatically. No one has to make any judgment. There's no bias. And what we find that all 3 doses produce a significant reduction in this, a reversion of the reduction in the paw luminescence, indicating that we are picking up analgesic efficacy. And then on the other side, we're using the standard technique, which is called von Frey mechanical testing, where we have a number of filaments that have different forces. And when the filament bends, you know how much force is being applied to the skin. And yes, indeed, we start off, you need a 1 gram force to get the animal to withdraw its paw. And when you have sinus, it drops down dramatically. So this technique does pick up the presence of mechanical hypersensitivity. But when we test the ketorolac at exactly the same drugs, we just cannot see a significant analgesia. So what I think this is revealing is that the standard approaches are too insensitive, and that's why you need such high doses to get an analgesia, whereas this new device with the pole luminescence is giving us a very clear signal automatically. Another standard model that is widely used by industry is the formalin model. Formalin is not only a fixative, but when you inject into the skin, it activates nociceptors directly through the TRPA1 channel, and it also produces inflammation. And it produces 2 phases of changes in behavior that have been picked up and are measured in 99.999% of people who use this assay. There's a phase 1, which is immediate flinches made by the animal after the injection. Then there's a quiet period for a few minutes, and then a second phase. And the way that you pick up this behavior is you take video recordings of the mouse after the injection, and you have someone sitting there with a stopwatch just counting how many times the animal makes a flinch or not. What we have now done is shown that we can do the exact same assay and look at changes in pore luminescence, and we get exactly as expected, an immediate reduction in pore luminescence, then some recovery, and then a second reduction as indicated in red. And when we give morphine at the standard dose, that is completely eliminated. So again, we think this is a technique that has an enormous advantage. So one of the things that OMA has done is to enable us not only to pick up the change in the pore surface contact, but to look at the behavior of the animal. So that if one gives an injection of formalin, as I indicate, there's flinching. And now OMA has devised a machine learning algorithm that very accurately detects the presence of different kinds of behavior. And this, he can differentiate licking and biting of the paw with flinching and other changes, and all automatically. And if you compare what a human individual does to identify these behaviors versus his algorithm, his algorithm is actually better. And he's called it ANI-ML, as you see, machine learning in animals, so animal. You're supposed to laugh. So this is now enabling us, when running these behavior tests, to pick up a whole range of behavior. And this is just a fraction of it, but we can look at the animals locomoting. We can see if it's standing still. We can see if it's biting its paw. And then there are the other forms of behavior which we can analyze. But just as you can see that in a naive mouse, you just put it in the box, it runs around, so there's lots of locomoting. There's periods where it stops moving, but not many. And there were only 2 paw bites over the full 30 minutes that this was done. If we then take a single animal and do this intraplantar formalin, the locomoting pattern has changed dramatically. If we had over there, you can see there was constant locomotion while the animal explores, but now it's very patchy, and there are prolonged periods where the animal is not moving. And that is fascinating. No one has ever before said not moving may be a readout of the presence of pain. But if you think about our own experience with pain, that is often a feature that if we have severe pain, we also don't run around, we also remain still. And then you can see the paw biting, we can pick that up automatically. And whereas there were only 2 paw bites in the naive animal, there are now a very substantial number. So we are gathering enormous amount of information. And then we can also look at the weight distribution. Initially, we only focused on the difference between the 2 hind paws, but recently we started looking at the difference between the weight bearing on the hind paws versus the forepaws, and all of those are giving us really exciting information, all automatic. So we can now run many assays. Our team can literally do multiple tests every day, And as I'll show you in a moment, that our major assay only takes 3 minutes. So we have turned our in vivo an analytic platform into a relatively high throughput way of testing for efficacy. And just as illustrated here are a number of animals with this analysis that I've described. And you can see the naive state, you can see the formalin state, and once again, you can see if the top line in each of them is the locomotion, you can see how it reduces. And then when you give morphine, you do reduce the paw biting dramatically. So there is an analgesic effect. But if you notice as well that the top line, which is the locomoting, is stronger than naive. The standard dose of morphine that is used in mice turns out to have massive side effects, including causing hyperlocomotion. So this new approach not only enables us to measure efficacy, but also adverse effects with enormous sensitivity. So while we're trying to test for our nociceptive silencers, one of the models that we've come up with that we think is going to be most useful, better than the formalin model, because formalin is pretty messy, which is to inject capsaicin, the pungent ingredient in chili peppers, into the hind paw at a very low dose. For those of you who like hot chilies, you know the burning sensation. That's exactly what we do. We're getting a burning sensation in the hind paw. And what I'll illustrate here are the behavior of a naive animal, just exploring. We've managed to keep we can take the animal and keep its body position fixed. And then on the other side, there's the intraplantar capsaicin. And what I want you to notice is both the behavior of the animal and the relative surface contact on the injected paw, which is on the far side. Because the beauty of this technique is we're not taking a snapshot, just one recording in time. We can record for over extended periods, which is giving us much more information. So here, this is a slowed down version. In the naive animal, the surface contact is indicated by this purple is equal on the two hind paws. But in the animal with the capsaicin, you can see there's a reduced surface contact. And it's pretty obvious here, and our paw luminescent signals pick it up very clearly as well. And it's not completely eliminated. We're giving a low dose, but it is significantly less, as illustrated here. And the animal is now biting and licking, and we can pick up that behavior as well. And one of the things that when we gave the injection, people, again, the literature is full of people applying capsaicin in various models, and almost always they measure for tens of minutes. But when we did our paw luminescent signals, we discovered this reduction in paw sensitivity, this increased sensitivity, such that there's a reduction in force on the surface, only lasts for a minute, because it's a very transient signal. And we can pick it up in this way. We can see exactly, and we can identify, are there gender differences? It turns out the behavior of both male and female mouse is absolutely identical. And we can then identify whether is this assay really helping us identify nociceptive silencers. So one way of doing this is to eliminate nociceptors. And one way to do that is to use a Cree driver of a gene that is expressed in nociceptors, which is TRPV1, which responds to capsaicin. And that then expresses diphtheria toxin, which kills off the nociceptors. So we have a way of selectively killing off nociceptors. And if you do that, and you then do this capsaicin model, the behavior completely disappears. So this form of behavior with the reduced surface contact does not manifest, and nor does licking and biting and flinching in the absence. So capsaicin is activating nociceptors. And then we can use it for drug screening. And here is just again showing morphine, where at a relatively lowish dose, 3 milligrams per kilogram, there is some reduction, and the complete elimination at 10 milligrams per milligrams per kilogram of morphine. If any human had an injection of 10 milligrams per kilogram of morphine, they would immediately stop breathing. That is a toxic dose to humans. So the dose of morphine that is effective in mice is very, very high. And we are looking for compounds that do not have this major toxicity profile. And this is the beauty of this approach. Unexpectedly, we designed it in a means to be able to interrogate what we call the body language of pain, but it's also picking up side effects. So here we have a naive mouse just exploring the little box, and you can pick up exactly what it's doing and where it's doing over time. And then we give two standard analgesics that are used in the clinic. One is amitriptyline, which is both a sodium channel blocker and an amine uptake inhibitor. And you can see that this is substantially reducing the movement of the animal. Essentially, the animal at the standard dose that produces analgesia is heavily sedated. And if you ask patients who get this drug, they also say they're sedated. They feel sleepy, they don't feel right. And then, as I mentioned before, morphine produces hyperlocomotion, and you can pick it up extremely easily in this. So that, again, most of the standard studies that use morphine just put an animal in, restrict its movement, they measure whether the animal's response to a noxious stimulus is affected. But in the meantime, if you leave the animal free, you see this massive change in its locomotion. So we can pick up all these potential adverse effects. And we are now using this platform to take the best hits from our screen, and we are finding compounds that do reduce the luminescence. We are finding compounds that reduce the pain-related behavior in the mice. So in the vehicle, you can see the mouse is biting and licking its paw that was injected with capsaicin, whereas with one of our hits, you can see there's a substantial reduction in that behavior. And Alex has put together a platform for looking at multiple features of mice behavior. We can do open field tests, we can do object recognition, we can do locomotor gait analysis, static posture, and then multiple features of particular behaviors such as rearing, grooming, etc. And also, in collaboration with Bob Data, we'll be using unsupervised learning, using a MOSIC type approach to dissect out behaviors that we may not even see or know about that may be present in particular states. Omer is not only helping us pick up these behaviors, but he's very keen to try and use this to identify potential biomarkers of pain. And he's doing a study now whereby he's doing freely moving behavior, picking it up with this platform we've got, but also making EEG recordings and other recordings from the brain, such that we can identify that when the animal does have a pain-related behavior, are there signals that we can pick up? And if so, can we then use them in patients? For example, a newborn baby, when it starts crying, it may be crying because it's cold or hungry, bored, or in pain. And we have no idea, none whatsoever. But if we had an EEG record such that there was a signal which always correlated with the presence of pain, that would enable us to identify pain without the individual having to report it. So I hope I've given you a flavor of the approaches that we're adapting to try and understand and conquer pain. And this is the goal of my lab. And you better work hard today so we can cure it tomorrow. Thank you. Yep.
It's a phenomenal system you have established with your extended team. I'm wondering, what do you consider the rate limiting steps right now that you want? Like, what are the major challenges that you would like to see move to the next level?
At this particular moment, it's the medicinal chemistry. So we get a promising scaffold, and then medicinal chemists look at that, try and see whether they can also interpret the interaction of the compound with the particular target, and using three-dimensional analytics. But in the end, they have to make multiple analogs, which we then have to retest and identify which ones look the most promising. But at least now we feel that we have a means to pick up, at least in vitro, the most promising compounds, so that when they go through a round and we get the new analog, we push them through our tests, and the profile we feel there is a profile that we feel, and so far it's beginning to work out, are predicting in vivo efficacy and some safety. So it's more complicated than just focusing on one thing. It's embracing the complexity, but in exchange for that, hopefully we are getting something that's going to have a more reliable predictor of activity in patients.
And just one other short question, and that is, what happens to the compounds that don't have conserved activity in mice? What are you doing with those?
Well, that's a fascinating question, because as I indicated, Vertex took the decision that they continued to develop a compound because it works with such high potency on this particular sodium channel NaV1.8. But it becomes very difficult, because do you know whether it's going to work in neuropathic pain or inflammatory pain or what? And they are spending literally hundreds of millions of dollars to find that out. We hope to get a much better sense of that, such that we can define which are the most likely clinical conditions where a particular therapeutic is likely to be most beneficial. And to do that, we need our mouse studies. So we keep the hits that do work well on humans, but we don't have the hundreds of millions that Vertex have to develop them. So we've shifted our focus to try and include those that do have a mouse. What we are also doing, which is another strategy, and hopefully we'll know in about a month's time, we are making mice that express the human targets as well. So we will have mice where we'll be able to pick up exactly this, but instead of having murine targets, they'll have human targets. So so that may be a way of moving things forward as well.
So on Zoom, someone just asked questions I think you just got into answering, but in case you have anything else to say, they asked if you can comment on recent advances in non-opioid analgesics, especially the Vertex mentioned. Is there anything else?
Well, so Vertex, being a pharmaceutical company, doesn't share. Frankly, we have made their compound and have tested it. But if you look in their papers, they don't reveal the compound. We've had to do some look at their patents and from that identify it. So they give limited information. They have had a New England Journal of Medicine article on using their VX548 in acute post-operative setting. And then they've had two reports, one in December and one in January, on phase 3 trials that have. But frankly, the amount of information in those is so limited that it's so difficult to know. What is clear is that there is analgesia, there seems to be safety. Frankly, it doesn't switch off the pain. It reduces it significantly. And in fact, when they compare it against standard drugs, whether it's pregabalin for neuropathic or a mixture of an acetaminophen and a weak opioid, it is as strong as their comparators. So they are hopefully making a safer drug that will have some efficacy, but it's not. What we want is to switch off pain.
So have you actually found compounds that respond differently for different kinds of pain? There may be a certain extent of heterogeneity there, like something that responds inflammatory.
Yeah, so the particular assays that I've shown you here were designed to identify nociceptive silencers, and that's why the in vivo platform that I described is we activating nociceptors and seeing can we suppress that. But we do have other assays that are looking for the effect of immune ligands on nociceptors, and they are changing their sensitivity. And that is ramping up and will be started. And we will, as I mentioned, we are studying neuropathic pain as well. That takes more time. There's a limited number of animals, but when we get the most promising compounds, we can test whether the compounds that we're discovering here also act in those conditions. But whether we can ever get an in vitro system that can mimic neuropathic pain in vitro is something which George, who I mentioned at the beginning, it's just a minor challenge that we're given.
Any final questions?
Do you think NaV 1.7 is a final common pathway? I mean, in terms of patients that have inactivating mutations that don't have pain, do they have some types of pain and not others? Or do you think that that really is a convergence point for different types of pain?
So the NaV 1.7 is another voltage-gated sodium channel, and it was one that caused enormous excitement in the field because there are patients who have loss of function mutations with congenital insensitivity to pain, and also gain of function mutations where the patients have a variety of spontaneous pain conditions. And based on that, the industry went crazy. Pfizer, Merck, Amgen, all of them attempted to develop NaV 1.7 blockers. And so far, not a single one of them has worked at all. They do work in patients who have gain of function mutations. They do reduce it. But I think what that has revealed unexpectedly is that at that time, industry for a while said they won't develop any drug unless there's human genetic data validating the target. And they said, this is the perfect validation. Well, it turns out when you knock out the function of a gene early in development, then there are compensatory changes. And I think most of the phenotype that we're picking up are compensatory changes rather than due to the actual mutation itself. So whether NaV 1.7 is going to be a major player, well, so far it hasn't been at all. It's a major cause of failed trials, actually. But one of the arguments is, has there been adequate target engagement or not? And none of the trials actually measured that directly.
Unfortunately, we're out of time. Thank you so much. Right now for the lunch, we're going to